psychology. Okay, so hormones would be the next topic that you would look at, and with hormones there's not much of a theory. It's just the basic idea that human beings have the same endocrine glands that animals have. Okay, they also produce um, oxytocin, they also produce vasopressin, they also produce adrenaline. Okay, so then because we have similar biological functioning and endocrine glands, their assumption is that we would have similar effects on behavior. Okay, so I'm not going to go over researches for brain and behavior and hormones and behavior because we are familiar with a lot of these. You will be working in groups of 3, okay, for this lesson, and you will brainstorm and you will write down in your either you can create a Padlet for your group, or you can create a document where you'll be doing an evidence bank for the animal extension. Okay, and these are some suggestions. You're not limited to these. I know there's Carl Lashley's study as well that could fall into brain and behavior. What I'm going to really go into is genetics and behavior because we haven't really looked at the use of animals in genetics and behavior. So I'm going to go and do that quite thoroughly. Okay, so the reasons why we use animal models in genetics and behavior is quite interesting. Okay, to begin with, there's really high genetic similarities between animals and human beings, and there's also the idea that we have evolved from a common ancestor. So if we have such high genetic similarities, then it is likely that our behaviors should be similar as well. Okay, and even with roundworms and fruit flies, we share 60% to 35% genetic similarity. Okay, so the argument is that because we have similarities in our genes, our behaviors might also be relevant to be studied using animal models. Okay, so one very good reason to use animals in our understanding or research of genetics and behavior is when we observe a polymorphism, which is basically a mutation, okay, in the human genome. So if, for example, we sense there's a mutation with the 5-HTT gene, okay, and we find this out in the human genome, then we would be able to then use very highly controlled variables and even modify the animal's genes on a molecular and genetic level by using knockout techniques, okay, and introduce this mutation into that animal and study it, okay. So we start off by studying a polymorphism in the human genome and then try replicating that in animals and then bring it into the lab to study what impacts that has on behavior, okay. And because we can't control the environment outside labs, again, you cannot bring human beings and keep them in labs forever, and you cannot control the history they have and the environment they had in the past, okay. So we then do that with animals, and because we're able to control the environment and control the variables, we're able to come up with cause-effect relationships between genetics and behavior, which we know is not really possible with all the research that you have come across on depression. It's highly correlational. We cannot ignore the third variables, and we cannot even ignore if there is bidirectional relationships. So use of animal models for understanding genetics and behavior really brings in the ability to create or establish cause-effect relationships. And the final thing is, a lot of animals have really short lifespans, so about 10 to 15 years. So then that means that we can observe different generations of animals and see whether there are genetic variations and whether these mutations continue to have effects over generations or not. This is not possible for human beings because we have an average lifespan of about 70 years. So that is another advantage of studying animals or using animal models in the study of genetics and behavior. So the first research that I will be discussing with you is Cases et al.'s 1955 research, and they used transgenetic mice, which are mice that have been artificially modified at a genetic level to include a foreign gene. This foreign gene is usually a human gene which is inserted into the mice genome. Like I said, there could be um, the 5-HTT gene that you observed in the human genome. Now you want to modify and manipulate it into the mice to see the effects of it in a lab setting. Okay. Brunner in 1996 studied a Dutch family who had several male members who were prone to impulsive behaviors, including aggression, abnormal sexual behavior, and even arson, which is having possession of, I think, guns. Okay. So the researchers sampled genetic material from this family and found a variation in the gene that produces monoamine oxidase A, an enzyme that affects neurotransmitters such as serotonin, dopamine, and norepinephrine. So the MAOA gene was often referred to as the warrior gene. Okay, so now that they'd found this kind of variation in that family, they wanted to bring this variation into the lab to see the effects that it has on behavior. Um, so when this variation results in low levels of MAOA enzyme, it increases the levels of the neurotransmitters that I just mentioned. So if it's increasing these neurotransmitters, does it have any effect on behavior? Okay, so Cases et al. Basically tested this variation in mice to check if similar behaviors may be portrayed. Similar behaviors being aggressive behaviors as seen in the Dutch family. Okay, they used the knockout method to remove the gene that regulates the production of MAOA. So from the mice that they used, they knocked out the gene variation resulting in an MAOA deficit. And the initial observation that they had was that for the mice that were between the age of 11 to 16 days, the signs were of low MAOA was of having frantic running, violent shaking during sleep, and a tendency to bite the experimenter. Okay, for the adult rats, they also observed aggressive behaviors, but these were more offensive aggressive behaviors, including bite wounds that were observed on different rats. Okay, so this was the initial observation, but they really wanted to see if it was just this or was it further aggression that they are capable of showing. Okay, so what they did was they introduced a resident intruder test. Okay, this test or procedure involves introducing a new mice into the cage of another mice. Okay, and now they compared two groups for this. There was the transgenic mice who had the knockout procedure, and then there was normal mice that were the control group. Okay, so when they observed the normal mice, they found that these mice merely just went and checked out the new mouse that had been introduced into the cage. They would sniff them and then they would engage with the new mouse. Okay. On the other hand, the mice with the variation, okay, they would react in a very threatening hunch, and they would also be very aggressive towards the new mouse that has been introduced into the cage. So these were very outward observations that they made of these animals, but following that, they conducted autopsies of their brains, which showed that there was an increase in serotonin, dopamine, and norepinephrine. Okay, dopamine was not as much, but serotonin levels were found to be 6 to 9 times higher than the control group. Okay, so the researchers then concluded that MAOA deficit led to strong aggressive behavior. But the thing is, human males who have this variation, their aggression cannot be attributed just to social factors. Okay, the MAOA deficit is a predisposition and can cause aggressive behaviors when interacting with social and environmental factors. Okay, we know that genes are only a part of the cause or part of the contributing factor to a certain behavior. So we cannot, even though they're able to establish cause effect in this experiment, we cannot translate that into real life or generalize that into real life because it's not necessarily ecologically valid to human beings. We have very dynamic environments. It is not necessary that every single person who has this variation is going to show aggressive behavior. Okay, it's just a predisposition that, if interacted with a certain environment, can result in aggressive behavior. Okay, and in fact, a majority of people who have this variation have found to be not very aggressive. The way that this research has found mice to be aggressive. Okay, this also makes us question the validity of how they have observed aggression. Human beings are capable of showing aggression in different ways. It doesn't have to be physical aggression. It doesn't have to be biting. It doesn't have to be punching. Okay, so it can be aggression in different ways. So we don't know if the MAOA gene is going to result only in physical aggression, which is again a major drawback. Since again it was a lab experiment, it does allow us to develop a cause-effect relationship, but again it goes back to seeing whether this is truly valid to human beings. And another major point to consider is that the results have been validated or have been found to be similar to that study conducted by Caspi et al. In 2002 on the MAOA gene and aggression in human beings. So there is triangulation, which goes on to say that the results are reliable and that here the use of animal model does in fact contribute to our understanding of MAOA genes and aggression. Okay. On the other hand, there's the whole issue of ethics. The mice were not euthanized, but they were autopsies, which basically means that the animals were killed. Okay, this is undue stress and harm, but also the procedure of introducing new rats, the resident intruder, and making them exposed to the aggression of the variation rats is also inducing undue stress and harm. So there is undue stress and harm in both parts. And if you look at it in the bigger picture, right? Let's say, for example, they had used more refined procedure apart from doing autopsies. Would we have been able to release these animals back into their natural habitat? They could be aggressive towards other mice around them and in their environment, but also now this would mean that the MAOA gene that we have artificially knocked out would be passed on to other mice, creating more aggressive mice in their genome. Okay, so there is kind of like a moral dilemma or ethical dilemma that we have with regards to this research. So the question is, was it better to use autopsies in this case because these rats would not be in a position to be sent back into the environment? The harm of doing that would be much greater than losing out again on a few mice. So, I completely forgot that I did this slide.